So we come to the last presentation of the session. It's great, I'm seeing some questions coming in. Please keep them coming. It's a pleasure to be able to introduce our final speaker, Dr. Melissa Sharpe, who's in the School of Psychology at the University of Sydney. Melissa's work explores how animals build cognitive models from learned associations, and she has a particular focus on role of the ventral tegmental area in modulating learning through dopamine signaling. And she's going to present work which characterises how animals guide their choices through differential weighting of different cue types during task performance. Mel.
All right, so thanks everyone for being here. This morning I'm really excited to be a part of this symposium. I'm actually doing rodent work, so I'm a little bit of an anomaly, but we've really taken our inspiration in the work that we're doing from the human cognitive neuroscience literature. So I'm hoping to draw some parallels between the work here at this conference and in the symposium, and to really get some feedback from all of you to see what you think about what we're doing and whether you think it's interesting. Thanks to Jason and Dragan for inviting me to give this talk this morning. And what I'm going to be talking about today is specifically a cognitive process that we've revealed within the lateral hypothalamus, where this region seems to be biasing learning and behavior towards cues that are most proximal to rewards and actively opposing learning and behavior towards cues that are more distal to rewards or irrelevant to rewards. The fact that I'm talking about the lateral hypothalamus as involved in this kind of reinforcement learning process might be a bit surprising because we typically think of this region as important for feeding. So the idea is if I turned on your lateral hypothalami right now, you would start voraciously eating anything that's in front of you. But actually, we showed in 2017 that it's doing something more than this. For example, it's really important for learning about the predictors of food in the environment. So being able to navigate towards things that are close to food so that you can consume them and survive. For example, in one experiment, what we showed is that we could optogenetically inhibit lateral hypothalamus, and specifically the GABA neurons that seem to be important for these learning effects, and really completely wipe out Pavlovian conditioning, and that is the ability to learn that there's a cue that predicts arrival of food. And specifically what we did was to optogenetically inhibit these neurons just during presentation of a cue that preceded delivery of a food outcome so that we could leave feeding intact but disrupt potentially the association between these two things. And indeed what we found is that we stopped these animals from being able to subsequently use this predictor of food to drive their motivated behavior to go and seek out that food. And we found that this was also the case when we weren't inhibiting these neurons. So it was really changing the way that these animals are able to learn about the predictors of food. And then in 2021, we showed something a little more surprising, which is actually that this region seems to oppose learning about more distal predictors of rewards. And we did this in a variety of ways, one of which was essentially to take a food predictive cue and present another novel cue that is now going to predict the arrival of that food predictive cue. And as we did in our prior experiments, we just inactivated this nucleus during presentation of that novel stimulus and not during presentation of the food predictive stimulus. And what we found here was that we actually enhanced learning about these associations. And what this suggested to us was that the lateral hypothalamus is this region that is really important for explicitly biasing learning and behavior towards those really proximal cues, but actually actively biasing away from those cues that are further from reward. And this is actually the only place in the brain where we see this dissociation. So some brain regions care about food cues, some care about cognitive maps that involve neutral associations, but really this is the only place in the brain that you inactivate and you can bidirectionally modulate learning in this way. And so I really wanted to develop a task in the lab that was going to allow us to look within a single session at how rats are able to use distal and proximal cues to make decisions, and we wanted to be able to inactivate lateral hypothalamus during both distal and proximal time points. And so the inspiration that we took for this was from Nathaniel Dawes' very well-established two-step task. And we worked with Nathaniel and also Marcelo Matar and Masakazu Tara. He's a postdoc in my lab. He was at UCLA and came over with me when I moved the lab 2 years ago to the University of Sydney. And we worked together to establish this task and be able to test in rodents the influence of more distal and proximal cues in learning and decision making. And so I'll take you through the basic idea of how this task works. This is one version of many versions of the DOOR two-step task. It's my favorite. It's the kiddie version. So essentially what they're trying to do here is to get to planet treasure. And these aliens down the bottom there in the red and purple, one of them is going to be able to take you to planet treasure. So you have to navigate through the first and second steps to be able to get to the right alien so that it can take you to planet treasure. So first you'll be presented with a first stage choice. This is what I think of as the distal choice, right? The distal predictors. And you need to choose whether you're going to go on the green or the blue rocket. So let's imagine that you choose to go on the green rocket. It takes you most commonly 70% of the time to these red aliens. So you choose one and it takes you to planet treasure. This is great. So next time when you're faced with that stage one stage, essentially the choice, you're going to make the same choice and choose the green rocket because it's going to take you most likely down to the red aliens and to planet treasure. But let's say that you make that choice on the next trial, you choose the green rocket, but surprisingly, it takes you down to the purple aliens. So you have to make a choice here. So you choose one of these aliens, and actually now you're going to get treasure again, and you get to go to planet treasure. So this might suggest that now this purple alien is the one that is leading you to planet treasure. So next time, actually, you're going to change your choice. And instead, now you should make a choice on the next trial to go on the blue rocket that's more likely to take you down to the purple aliens who predict that you're going to go to planet treasure. And so essentially the idea is that you need to use the transitional structure of the task in order to inform your choice on the next trial. And if you're doing that well, then the data should look a particular way. And so what I'm going to show you here are the stay probabilities of how people usually respond. So if you see higher numbers, they're staying on the next trial, they make the same choice that they made. Or if you see low numbers, it means that they're going to switch. And so, for example, if you were to go to the green rocket, you go to the red alien, you go to planet treasure, and you essentially want to make the same choice, so you're going to stay. But if you had that rare transition and you go surprisingly to the purple aliens when you choose the green rocket, then next time, if that takes you to planet treasure, you should choose the blue rocket. Does that make sense? It's a complicated task. Hopefully I took it through you well enough. And you should see the opposite when they're unrewarded trials. If you didn't take anything away from what I just said, essentially we're just looking for this U shape, and that's showing you that people are tracking the transitional structure of the task to inform their decision on the next trial. And so here's our rodent version of the task. So they're at start by initiating. They go into a port and they say, okay, I'm ready for a trial. And then we give them what we think of as this distal cue or the first stage choice. And so they essentially get given either the green cue or the yellow cue. And once those are finished, the levers come out and they need to make a choice. And once they've made a choice, it leads to these probabilistic transitions towards the proximal cues. So only one transitional structure is going to be relevant on a trial. For example, if you were to get the green cue at the distal point, you need to make a choice. And if we're trying to get the animals to find that pink proximal cue that's going to be rewarded, they're going to choose the left lever there. But this could surprisingly go to the blue proximal cue. And if that leads to reward, this means that they need to change their behavior on the next trial, and they essentially need to press the right lever when they're given that green cue. And so essentially, when we simulate these data, it gives you the same kind of U-shaped function that tell us that those animals are capable of thinking back through the transitional structure of their task to inform their choice on the next trial. So again, we're just looking for this U-shape that tells us that the animals are able to do this. And so the animals actually perform really well on this task. So they're capable of adapting their behavior as the contingencies shift. They have to reach a criterion of 70% before we start doing these reversal changes of whether the reward probability shifts on the basis of the proximal cue, so we're changing which proximal cue is rewarded, or the distal cues are shifting, so there are different contingencies in effect. But they're really good at adapting their behavior across these 2-hour sessions, so they invoke a number of changes across time. So that's all well and good, but are they actually using the transitional structure to inform their choices? And the answer is yes. So if we analyze the data just around these proportion where they're getting close to the threshold to initiate another reversal, you can see that we see this really nice U-shape that tells us that the rats are capable of thinking through that transitional structure to inform their choice. And how we analyze this is to use logistic regression. So essentially what you're looking for is that interaction between the transition, that's the identity of the cues that occurred, by the outcome, whether or not they were rewarded, and that indicates that the animals are responding differentially depending on the transition that they experienced and whether or not they were rewarded, and so that U-shaped function is meaningful. So rats are able to use this transitional structure. And so now that we've got that working after about 3 years, we're able to use optogenetics to inactivate different neuronal populations at different time points. And I just really want to emphasize how it is that this task allows us to really differentially pass the role of neural regions in very different time points during reinforcement learning. And so here's the task, and essentially the reason that we designed it in this way is that we want to be able to inactivate during the distal cues, or the proximal cues, or the outcome. And optogenetics, of course, really affords us very tight temporal precision on neural activity so we can inactivate across these very short timescales. And so the data that I'm going to show you today, even though we developed the task essentially to study the lateral hypothalamus, and Master is currently running these experiments, hopefully not on the weekend, but he was certainly running it yesterday. But what we did first was to kind of validate that we're actually generating the cognitive processes we think we are by testing whether the orbital frontal cortex is important for navigating through the transitional structure of this task. And so, of course, we know that we form cognitive maps of our environment, and that the orbital frontal cortex is important across species in both rodents, primates, and also human primates. And what we wanted to do was to test that rats are really using these kinds of cognitive maps of the world to influence their behavior. And how we did that was to put a virus that's carrying a halorhodopsin into the orbital frontal cortex, which allows us to control the neural activity of pyramidal neurons in the orbital frontal cortex with light. So we inhibit their firing over these very short timescales by delivering right into the brain as these rats are moving around, making choices, and going and collecting rewards and experiencing the transitional structure. And so the first thing we did was to inactivate pyramidal neurons in orbital frontal cortex during the distal cues in the task. And this is our control rat. So we have sessions where we don't have optogenetics and sessions where we do shine the light. And these are our control rats. They have a virus in the brain, but we're not inactivating OFC. It's a control virus. But the light goes into the brain, and they've got all the hardware and a virus, right? And you can see their behavior looks really nice right before the optogenetics session. When we shine light, it doesn't disrupt their behavior. And you can see in the logistic regression, we get the nice transition outcome interaction. And we don't see an outcome by opto, which is essentially the session, so we're not disrupting behavior by shining light into the brain. But then if you look at our MPHR group, where we are inhibiting these neurons with light, you can see that we significantly disrupt the ability of these animals to use the distal cues to inform how they're going to make choices on the next trial. So they're no longer using the transitional structure of the task to inform their decision-making. And you can see what they do instead. You can see we see this statistically in the interaction with the session, the opto session. They stay if they're rewarded, so they make the same choice, or they shift if they lose. So they just adapt a win-stay policy, and they are no longer using the transitional structure to inform their choice. So optogenetic inactivation of OFC during just the distal time point disrupts their ability to use transitional structure. And so then we wanted to see what would happen if we inactivated during the proximal cues. And so here again are our EYFP controls with and without the light. Here we actually saw a little bit of an effect of the light, but it didn't change the significance of the U-shape across each session, so they're still showing use of the transitional structure. And when we inactivate OFC, so in our MPHR groups here, we see that we don't affect behavior at all. So this suggests that inactivating the orbital frontal cortex during the proximal cues doesn't disrupt their ability to use the transitional structure of the task and change their response on the next trial. And so this suggests that OFC is not just encoding cues, right? It's specific time points at which it's really important for behavior. And the final thing I'll show you is inactivating orbital frontal cortex during presentation of the outcomes, reward or no reward. And so here's our EYFP group, no effect of light here. And our MPHR group, again, we find significant deficits in how these animals are able to use the transitional structure of the task when we inactivate orbital frontal cortex. And again, they seem to adapt a bit of a win, stay, lose, shift policy here. So this shows the optogenetic inactivation of the OFC during the outcome period reduces the use of the transitional structure of the task. So I hope what I've shown you today is a new two-step task that is allowing us to dissociate the neural circuits that are involved in proximal and distal learning and behavior. We found that inactivation of OFC only disrupts behavior if we're inactivating at the distal or the outcome point. So it's not just important for any aspect of the task, we're really dissociating what it's important for, and we're currently doing the modeling to try to understand how it is that OFC might be influencing behavior on the task with Marcelo. And essentially, this is demonstrating that we're recruiting areas that are important for navigating through cognitive maps and developing cognitive maps using our task. Now we're looking at the role of the lateral hypothalamus in modulating behavior between distal and proximal cues, and this will constitute the first formal test of the involvement of lateral hypothalamus in reinforcement learning in this kind of way. And so I'm really excited to see what we find, and I really encourage anybody that's doing human work to think about this brain region as something that's important for learning. It's not just something that's doing feeding and sleep, right? It's really doing something important, and would be really excited to hear if anybody would like to work on something like that together. Just a shameless plug, I'm hosting Pavlovian in Sydney in August. These are just our invited speakers. We have about 50 speakers. If you're interested, come chat with me or scan the code there. And thanks to everybody in the lab for navigating what was a pretty big move between UCLA and the University of Sydney. And especially a big thanks to Marsa, who did all of the work here. He's a really phenomenal postdoc. He does all of the modeling with Marcelo as well. And to, of course, Nathaniel and Marcelo and funding bodies. Thank you.